In December 2025, a man in Australia did something amazing. His dog named Rosie was diagnosed with mast cell skin cancer, which was almost untreatable. The dog had become very weak. Well, he was not a biologist or any kind of involved in biology, but surprisingly, he made a cancer vaccine for his dog on his own using AI tools which are generally available. The injection, the therapy was successful and the tumor shrank by 75%. And that's what got my interest into this topic, the world of biology and AI, and that's what we will be talking about today. We will talk about what he did, how he did it, what tools he used, how AI is actually going to change biology and solve a lot of problems that we face today in terms of diseases and disorders. That's not all. We will talk about a little bit about negative things also. That's what makes a video complete. So let's start. It all starts with a dog named Rosie. It was a Staffordshire cross breed which he adopted from a shelter in 2019. But later, it was diagnosed with advanced mast cell tumor cancer in his hind legs, and the type of cancer, especially in dogs, is almost untreatable. No modern medicine or treatment is there to treat that kind of cancer. He still spent thousands of dollars on traditional chemotherapy and nothing worked. The tumor did not shrink. The standard Maxine and vets had given up and they said that the Rosie has only 7 to 8 months left. But the owner of the dog, Paul Cunningham, wasn't the one to accept defeat. He was a tech entrepreneur in Australia who had been working in the field of machine learning and data science for the past 17 years. He had zero biology degree or any kind of clinical training, and it was not reckless. What involved was a lot of hard work, researches, working a lot of extra time, all to treat his dog. In the end, he was an entrepreneur, the person who knew how to do things, how to make something from invisible and bring it to existence. Well, he did it. Firstly, he contacted UNSW Genomics Research Center, where they sequenced the DNA of the Rosie dog from both the healthy cells and the cancer cells, and it costed around $3,000 in Australian currency. Later, after receiving the cumbersome files of genes of the DNA, he used AI like Grok to make and divide them into various parts and diagnose and analyze the data set using Grok and compare the damaged tumor cell DNA with the cell DNA of healthy cells. After that, he fed the mutations into AlphaFold, where he identified which protein structures were damaged by the mutations and which drugs could counter them. Using all that knowledge, he designed a custom mRNA vaccine, which was targeting those specific mutations. Same core technology that was used for developing COVID vaccines in just 11 months. And this time, it was pointed at cancer. But the hardest part was not making vaccine. The hardest part was getting approval from the ethics committee. He wrote a 100-page ethics document which took him around 3 months and 2 hours every night just to get the approval to run the trial on his own dog. After that, a second scientist who volunteered to administer the vaccine to the dog injected it around the tumor. This technique is generally called immunotherapy, and the primary injection was followed later by a booster dose, and all of this actually worked. The tumor shrank a lot by around 75%. Second vaccine is now there for the remaining part of tumor, which is more stubborn because the cells in tumor all have different genetic mutations as they evolve much faster. So some cells were not targeted by the previous vaccine as they may have evolved further, but still He is trying to make vaccine for that second remaining part of tumor also. And it is not just for dogs or animals. The technology for mRNA vaccines using AI can also be used in humans also, about which we will talk more in later parts. And the thing is that it was not a random guess that person did. I mean, he was not even in biology field, but he did it. And what it tells us about is a very strong signal that biology is actually about to become more accessible. The people who really want to do things till now but didn't had resources or didn't knew how to do things but just had an idea in mind can do it now using AI tools. The expertise is there. Just what you need is a very good idea and the ability to learn more, follow more steps. The tools he used are already here and mostly free. ChatGPT is almost free, AlphaFold is free and publicly available. The genome sequencing costs are collapsing at an exponential rate, becoming way cheaper, and things are becoming way more accessible. So what changed was finally that someone who needed it most and wanted it most assembled all the things and used them in correct way to finally treat his doctor. But to really understand why what Paul did was so revolutionary, we first need to go back around 50 years to a problem so hard scientists said it never could be solved, and understanding that problem is now almost completely solved using AI. We will talk about all of that in the rest of the video. Let's go. Basic unit of everything is in the end protein. It is in our body and it runs everything: enzymes, hormones, antibodies, muscle fibers, receptors present on every cell. Even our immune system is made up of proteins in the end. And a protein is made from a single chain of long amino acids. The sequence for which amino acid will go where is encoded in our DNA, which is then transferred to RNA, and then RNA makes it into a protein. But a flat chain of amino acid doesn't reveal much about it, like its structure, how will it function, or anything else. It's not useful to us until it falls in a shape which can bind to anything or work with anything. And that 3D shape was the problem. Once the 3D shape is even slightly changed, the protein just stops working, or worse, it could start misbehaving. Most of the diseases like cancer, Alzheimer's, Parkinson's, heart diseases at the molecular level are because some random protein folded in a wrong shape, and it caused a complete malfunction of all the cellular machinery in our cells. For more than 50 years we could read the sequence of amino acids in a straight line, but what kind of protein structure it would make to predict that was very hard. Now this is the protein folding problem, which was formally recognized in 1970s. In 1972, Christian Efferson successfully proved that the final shape of a protein is determined entirely by its amino acid sequence. The information is present there, but we just cannot decode it accurately. But then came Levinthal's paradox, which was by Cyrus Levinthal from MIT in 1969. He calculated that a single protein can exist into 10⁻300 possible conformations, shapes, and that is amount is more than the number of atoms present in our universe. If a supercomputer was able to calculate a single protein structure every nanosecond, it would take it more than the age of universe to calculate all the possible shapes for a single protein. How many proteins are there that we know of? More than 200 million proteins exist, and that's what we know of. There could be billions, trillions more than that we are still unaware of. It was impossible to accurately predict the structure of a protein actually. There were some methods to do it, with which we could discover around 100,000 protein structures in last 50 to 60 years. Yeah, like it would take a guy's whole PhD degree to discover a single protein structure. Like the techniques were there, like NMR, nuclear magnetic resonance, or cryo electron microscopy, or X-ray crystallography. These all would take a long time to do and were very expensive, some of them costing around more than $100,000 for per protein, and for preparation for that would take around 3-4 years to find a accurate structure of just a single protein, and that was a bigger issue. And if you calculate like the cost to find the structure of 200 million proteins, it would cost us around $24 trillion. That is just too expensive for anyone to do or fund. Like most of the major economies are under that. And the reason that people need to find protein structure, like they are able to spend so much money, is because every drug on the earth that we use ourselves work by binding to some kind of protein. If you don't know the accurate shape of that protein, you cannot design a drug to work for that disease. Like it's like trying to make a key but you don't know what lock to fit in it. The key is basically useless, or you would have to try it in thousands, millions of locks before finding out the right one, which was a cumbersome process full of trials and errors, very expensive also. So to tackle this problem, CASP program was started, Critical Assessment for Protein Structure Prediction, which was held every 2 years, where people from around the globe came with the technologies that they had developed to find the accurate protein structures at cheaper cost and faster and accurately. And for 25 years there was complete stagnation. No one came close to finding the right things that they wanted at the competition. But Google in 2018 for the first time showcased and participated in CASP 13, where they showed their AlphaFold model. There the model came first, but the difference from the second and third participants, like winners, were not that big. It was just a marginal improvement, so nothing big was expected out of it. But then CASP 14 happened in 2020, and Google this time came with AlphaFold 2, a better version, a more accurate, improved version, and it defeated everyone there. The model had a median GDT score of 92.4 out of 100, which was considered as close to the experimental results could get it. Like something about 90 was considered phenomenal and impossible till now. The organizers called it the final solution to the protein folding problem. One researcher described it as a first-class scientific result. The whole community was stunned by it. DeepMind in one competition did what 25 years of human minds couldn't. And it was all done using AI. And due to this, in October 2024, like it took them 4 years to realize, but Demis Hassabis and John Jumper, both the co-founders of Google DeepMind, won Nobel Prize for that. Nobel Committee said that AlphaFold has finally solved the single protein folding problem. Essentially, it was a first time proof that they could use AI to solve everything, and starting with the protein folding problem. Now they have other projects also. So DeepMind essentially solved, like not completely solved, but helped scientists figure out the problem, one of the largest problems in the history of science. But the more important thing is how they did it. What is AlphaFold actually doing that no one else could do? Remember the part where I talked about using a supercomputer to find the every possible structure for every single protein possible? Well, AlphaFold doesn't do that. That's why it's much better in finding accurate protein structures. What it actually does is it uses a concept called coevolution as a shortcut for finding the protein structures. What happens in nature is, if two proteins have evolved from same ancestors and into different lines, then there is a possibility that they have similar sequences and similar structures. AlphaFold's job is to read 3.5 billion years of evolutionary history on the planet Earth, whole just as a map. To actually find the accurate structure of a protein, what we need to feed AlphaFold is just amino acid sequence, which is very easy to find. After taking that amino acid sequence, AlphaFold runs through it and finds MSCs, multiple sequence alignments, all the thousands and millions of relatives of the same amino acid sequence from nature. And then using coevolution, if two kinds of amino acids have evolved similarly, like if amino acid A and B always mutate together across thousands of species, it's possible that they are physically close to each other in 3D space. They coevolve because they physically depend on each other. AlphaFold reads these coevolutionary signals as a map of where the protein makes contact with itself in 3D. The neural net that it uses to predict structures is called Evoformer, and it is the core architecture of AlphaFold 2. A transformer, of course the same that we use in LLMs, the transformer based neural networks, but this time built only for this problem and fed only biological data. And while running the simulation of protein evolution, it just evolves and changes the protein structure consistently until it reaches to the right structure. After hundreds of these update cycles, Evoformer produces a deeply refined understanding of the protein's probable 3D structure. It's like a sculptor. Evoform first gives you a rough block, but with time it refines it further and further after taking the data and comparing it to other evolutionary partners and proteins which co-evolved along with it, about which it already knows, and then predicts the structure of the modern protein using past relationships. It also gives itself confidence scores to help scientists understand if the structure is truly accurate, and if AlphaFold model believes it to be accurate, then chances are that it is also accurate. Inaccuracy tests, RSMD, root mean square deviation, is used where the protein structure that AlphaFold predicted is compared with the protein structure experimentally found. In this RMSD, value of 0 means perfectly similar, a value of between 0 and 2 is almost good, and 2 plus means the protein structure predicted is completely different. AlphaFold's median RMSD score is 1 angstrom, which is highly accurate. Like for experimental results it's 0.8. So AlphaFold is actually very accurate at finding the true protein structures using just prediction. However, there were some limitations with AlphaFold 2, like it cannot predict the structure for multiple proteins which are all combined, protein complexes actually, or nucleic acids, RNA, or any other things. One more thing, the proteins are never static because they are in a fluid in our body. They constantly change their shapes, interact with different molecules, and that interaction and change in shape also cannot be accurately predicted by AlphaFold. What AlphaFold does is just predicts the core protein structure, not the molecules around it with which the protein interacts. That is a problem that was solved by AlphaFold 3. Another problem was that it still needs wet lab experiments to find the protein structure experimentally. The AlphaFold 2 only predicts the structure. The accurate structure is yet to be found using the same methods like NMR or X-ray crystallography to be used for real biology experiments or for real drug making. But it works as a guide. Earlier, scientists had to figure out manually for each and every protein structure, but now they have a map and probability. Like they know which protein structures are there and which will, if found the structure experimentally, work as a best and bind best with the target molecules. So it just acts as a guide. They now don't need to go through the trial and error expensive process where every protein structure had to be synthesized. They just know the structure, but for final use they have to find it accurately using experiments. But for limited proteins, then everything else. And to solve some of these problems, like the structure of RNA, protein complexes, and everything else. AlphaFold 3 was launched in 2024. It was a collaboration between Google DeepMind and Isomorphic Labs, also owned by Google DeepMind. It has now more complexity. It uses a different kind of architecture from evoformer to pairformer. It can predict complex protein structures, small molecule ligands, antigen-antibody interactions, and many more things. Everything that interacts with protein in biology can be predicted by it with high accuracy also. Another thing it added to itself was diffusion models. Instead of building a structure piece by piece, now it starts from a shapeless, blurred vision and finally perfects it layer by layer with time to reveal the final structure. Everything is now unified and it's 50% more accurate on protein molecule interactions than prior tools. And that is why Paul was able to solve the problem of cancer vaccine on his own using AlphaFold 3. The AlphaFold didn't just told him the shape of the protein, but it also told how it will interact, how the proteins will interact with the mutations in his cancer, and how the proteins will interact with the immune cells and force them to target the tumor cells actually. AlphaFold 2 was made open source and publicly available to use by commercial labs, but after the founding of Isomorphic Labs, AlphaFold 3 is largely closed source. Like the protein structures are publicly available, but they should not be used for commercial purpose and only for academic purpose. And the source code is also not released for AlphaFold 3. Oh, actually it was released later after the scientific community forced them and raised their voices from Google to release the structure. But in the end, the Isomorphic Labs has to earn money. Google forced more on its commercial benefits and it never released the newer models after AlphaFold 3 for public. Nothing about them. So now you know the basics of AlphaFold 3. For more deeper dive, you can check out the Veritasium video which he made around last year he made it. You can check out that one. But knowing a protein shape is step one only. The real question is what you can actually do with that other than making the vaccine, which he did. Specifically, how can it change how drugs are made? Because right now making a new drug is broken in a way that most people don't realize. It's expensive, it takes a lot of time, resources, and most companies just don't want to do it. And that's what we will talk about now. Currently, on an average, each new drug takes around $3 billion worth of investment and around 10 to 15 years to its succeed. Once clinical trials begin, 90% of the candidates fail in them. The success rate is just 10%, and after a massive investment, because nobody knows early enough the drug will work or not. Most waste happens in the early stages also regarding money. It is essentially a global scientific lottery with a very hefty entry fees of $3 billion. Very few can afford to lose. And that's where AlphaFold changes a lot of things at not just an initial stage of protein structure prediction, but at each and every stage, starting with target identification, where AlphaFold reveals the protein structures which are actually linked to a disease that we never knew existed or couldn't have found so soon. New structures means we can target them in a newer ways using newer potential drugs, and in the end treating that disease with novel methods. Another thing where AlphaFold helps is in lead discovery. Instead of synthesizing thousands of molecules blindly and testing them physically in the labs, researchers can now virtually screen millions of candidates against the protein structure. Only the best go to wet labs for final testing. AlphaFold 3 also predicts drugs binding affinity, metabolism, bioavailability before a single experiment is run. Even it can also predict the failures that could lead to It can predict failures also with the help of protein and molecule interactions, due to which drugs which are most likely to fail are killed in earlier stages before investing a very heavy amount of money in them. And it is already changing the real world. Researches linked to AlphaFold 2 are likely to be cited 2 times more in clinical trials, which means people trust the AlphaFold 2 times more than any other tool available. There is a 40% increase in novel experimental protein structure submissions after AlphaFold's release. Apolipoprotein B100, ApoB100, the central protein in low density lipids, i.e. cholesterol, had been structurally unknown for a lot of time, and this protein is what's responsible for the heart attacks in individuals. But now we know the accurate structure and how that complex works using AlphaFold 2. Another big was SPINK1 gene in Parkinson's disease, which was matched to its instant equivalent, and because that was highly studied by scientists, researchers could predict the human equivalent of that gene more accurately using AlphaFold. More relatives you have to a protein, more you can accurately predict the structure. Okay, AlphaFold 2 did all of these things. AlphaFold 3 was highly restricted for commercial use, but Isomorphic Labs didn't just succumb themselves there also. Their next big things, which people also call the generation beyond AlphaFold 3, was ISO-DDE. Full form Isomorphic Drug Design Engine, a unified computational drug system built on the basics of AlphaFold 3 but not restricted to it. It can predict entirely new protein ligand interactions, ones that were impossible for the prior AI models, and the training data has never seen them. It is 2 times better than AlphaFold 3 on significant benchmarks. Can model cryptic binding sites. No one else could. What are cryptic binding sites? Well, these are hidden pockets in proteins that only open under specific conditions, and these are impossible to find experimentally, as in most experiments the proteins are studied in isolated conditions. Like, it helped in finding the sites for protein cereblon. For 15 years, scientists found only one binding site in it. ISO-DE predicted both the existing one and a brand new one from the amino acid sequence alone, and no expensive experiment was needed to do it. Happily, well, Isomorphic Labs isn't the only one company that is focused on solving these problems or using this tech. There are many other companies like Axis Scientia from UK, which made OCD drugs which are currently in clinical trials, and anti-cancer molecules which are currently in phase 1, which is research phase. But surprising part was that all of it took from research to clinical trials just 8 months for OCD drugs. They use a DMTL loop, which is design, make, test, learn, while running the equipment and AI autonomously. The AI learns on its own, experiments on its own, learn from it, and design newer things, enhancing and accelerating the loop very faster. Another company named Insilico Medicine, which underwent world's first phase 1 trial for a drug designed by AI, which was for idiopathic pulmonary fibrosis. It completed all of the research in just 30 months, which earlier would take around 10 years, and drug also saw the possibilities for anti-aging properties in it. Another big thing was from IBM, namely IBM Watson. It found 5 new RNA binding proteins linked to ALS, a disease too complex for any human researcher to fully untangle. Also matched those patients to clinical trials using predictive algorithms to find the right matches for right drugs to reduce the failure rates dramatically. And all of it can be seen in the cost math of these companies. The implementing AI in early drug discovery has a potential 70% cost reduction, and at 70% reduction, drugs for rare diseases, neglected tropical diseases, and conditions affecting a very unique part of population can be discovered. The cost has decreased dramatically with which newer startups which don't have much money in the initial times to spend, unlike bigger pharma companies, can now take part in the race, making things cheaper, more commercialized, more democratized. Overall, good for humanity. What Paul did with Rosie was not something that traditional pharma companies do. He didn't find an existing drug or slightly modified it. He designed something brand new, completely different vaccine, and it was a personalized one according to his dog's genome sequence. And to understand that, we must now see to a different kind of technology called gene therapy, and everyone has actually taken the benefits of it during COVID. The vaccines which we took during COVID were primarily developed using AlphaFold and mRNA vaccine technology. Every person who got a Moderna or Pfizer vaccine during COVID-19 definitely received a mRNA based vaccine. The basic idea is that mRNA gives instructions to a cell to make what kind of protein. That protein then trains immune system what kind of cells to attack on, and that's how the whole loop works. But the same technology can also be used in another big problem of humanity: cancer. Cancer treatment can be made successfully using mRNA vaccine, where the cells of immune system are trained specifically to attack the tumors in our cells. The genes which have gone mad or have mutated, due to which they differentiate, work abnormally, and are not identified by immune cells because they are part of our body. But mRNA vaccines can train the immune system to attack specifically those tumor cells. Another problem is that in tumors, every gene, every cell has a different genomic sequence, different mutations, and no two persons cancer can have similar. It is possible that they have similar mutations, but it's rare because tumor cells mutate very faster and go haywire sometimes, which was not case with COVID, because everywhere the COVID virus had same genetic sequence. So cancer vaccines has to be personalized, and currently that personalization is very expensive. But with mRNA vaccine, the time will also be reduced and it will become cheaper and accessible to everyone. And that's the target: to design mRNA vaccines to teach the immune system to destroy cells, but faster and more accessible way. Whereas traditional vaccine development takes more than 15 years from concept to final approval, personal mRNA cancer vaccines like Moderna can be made in just 6 weeks per patient and in a mass scale, because the manufacturing process is actually identical for each of the patient, just the mRNA sequence changes. And this is also the reason why COVID vaccine was produced in just 9 months. It went from an idea to final FDA approved drug in just 9 months. The mRNA infrastructure already existed, and there is real clinical evidence to show that mRNA vaccines for cancer actually work, like how Moderna or Merck mRNA 4157V940, which was a personalized mRNA cancer vaccine for melanoma, the skin cancer, It is currently in phase 2 trials where it showed a 44% reduction in the recurrence of cancer cells. The same treatments are also in development for other kind of cancers like pancreatic, non-small cell lung cancer, bladder cancer, many others. Pancreatic cancer, which is the most lethal type with a 5-year survival rate of 12%, and mRNA shows significant promise to solve that problem also. And what Paul did was exactly this timeline: tumor sequencing, mutation identification. AI just predicted the neoantigen that was to be made for training the immune cells, and finally designing a custom RNA to inject that message into the body. That is the thing which used to take millions of dollars for clinical facilities to do. He did with just a small investment of $3,000 and 3 months of paperwork. The majority of time actually went into just doing the paperwork. The vaccine was actually ready much prior, and that shows that FDA and other authorities also need to be changing their standards and ways. And that is the story of how mRNA vaccines was made, how a person named Paul, who wasn't even into the field of biology, designed vaccines for his dog. Not only designed, the dog recovered a lot actually. Soon after it, the dog could run, which it couldn't do before. The dog felt a lot healthier, and that shows us how in future the medicines are going to change, how you would have personalized treatments, how cancer will change from something uncontrollable, deadly, to something which can be managed using science and AI. And one of the bigger leaps would be anti-aging technology, which is also being developed currently using AI in a completely different field. Of course, AlphaFold helps in that also, but it's just a little glimpse of the future. It seems very exciting to live in that future, sometimes sad. There is a paradox like what will happen. Let's be optimistic, but the tech is actually emerging very fast, leading there. Could one day lead us to a multiplanetary species that just never dies, or a species that, according to Geoffrey Hinton, really goes extinct in next 30 or 40 years. Who knows what will happen. Until then, see you, bye bye. My next video will be uploaded soon. You can subscribe to my channel, and of course, if you have any thought to share in the comments, and we will discuss it also. Check out my prior videos, and